Hello everyone, my name is Diffusion and I am continuing my neuroscience series, and today we're going to be talking about membrane transportation. And I have a bit of a change of scenery because I have moved back to college, and here is my little friend Tommy, and we're going to be creating this video today for you guys. Awesome. Let's get right into it, shall we? Cool. So the cell membrane, as we all know and love, is considered to have a phospholipid bilayer, right? Which means there's two layers to the cell in which both the layers are amphipathic, which means that's impenetrable to water-soluble molecules. So anything that can dissolve in water cannot go through the membrane by itself. This is why we have membrane-bound proteins, which means that the membrane itself will be semi-permeable. Depending on the protein at hand, you will be able to transport certain molecules through without having any issues. Now, it's a little bit harder when we come in terms of things like glucose, for example, because of its pure size. It's very large, and larger molecules are going to have a harder time fitting through these smaller membrane proteins. Like I was saying, we have membrane, excuse my squares, they're a little uneven, but we have the extracellular side, which is outside, and then the intracellular side. Now, keep in mind, the molecules that are present on both of these sides are very, very important to the transport that is about to occur. And I apologize for the change in light just now, my screen just turned off. So depending on the amount of molecules you have outside the cell, so let's say you have X amount of positive ions here. You have, let's say you have 5, and you only have 2 on the intracellular side. You're going to want to have molecules flow to this side to reach equilibrium, right? Basically evening out the sides and reaching what we will eventually get to know as our resting potential. So this type of transportation is done without using ATP, and ATP, as we all know and love, is a form of energy, adenosine triphosphate. So we're going to discuss 2 different types of transportation, which is active and passive, and there's going to be a couple subcategories within these. First type of transportation we're going to be talking about today is going to be our simple passive diffusion. Great. So to begin with, let's draw out our membrane again. Like we said, there's 2 layers in this membrane, and within this membrane, we have membrane-bound proteins. So let's just draw a couple circles, and these circles are going to represent our membrane-bound proteins. Sound good? Cool. So simple passive diffusion works in a way that no energy is needed, no ATP is needed, nothing like that. Basically, you're moving from your concentration gradient from a high concentration to a low concentration to reestablish what we know and love as equilibrium. Awesome. So like I said, we're moving from a high concentration to a low concentration. Let me give you an example. Let's say this is the outside of our cell and this is the inside. On the outside, we have a bunch of positive ions. Cool. And on the inside, we only have 2. This ion is out of equilibrium, just for this case. What do we do to make our ion go from being out of equilibrium to being in equilibrium? There's an imbalance. There's too many positives on this side and not enough on this side. Let's go back to our rule, high to low. We're going to move some of these guys from the higher concentration to our lower concentration. So they're going to flow through. Now, remember these membrane-bound proteins I was talking about before? Disregard them. They're not going to matter for this because simple passive diffusion doesn't have to worry about these guys because they don't utilize them. Now, there's a bit of a caveat to using simple passive diffusion, and that occurs because ions cannot be transported through simple passive diffusion. No simple passive diffusion. This is purely because of the size of them. An important thing to remember when talking about simple passive diffusion is Fick's law. Fick's law. And I apologize for writing all over the page, but it's a little more interactive for myself. So, Fick's law takes into account 2 major components, which are the membrane thickness and the membrane area. I'm going to go over this pretty quickly, but basically, the thicker your membrane, the harder it is going to be for molecules to go through the membrane. The wider the membrane, the more area you have, the more space, the easier it's going to be. So there's a positive correlation for membrane area. So this is, if I increase it, then the diffusion rate will also increase. Now let's look at membrane thickness. If I increase membrane thickness, my diffusion rate is going to decrease. Awesome. That's the basics of what you need to know for simple passive diffusion. The next type of transportation we're going to be talking about is facilitated diffusion. So, facilitated diffusion. Again, for this one, there is not going to be any source of energy or ATP. So there's three types of this facilitated diffusion. We have pores, channels, and carriers. And carriers are also can be named uniporters. But let's start off by going through the initial differences, then we'll go into what they actually look like. So pores have 0 gates or channels. Let's stick with the word gates because it's going to get a little complicated if I use channels and then talk about channels. For channels, you have 1 gate. Carriers, you have 2 gates. Awesome. So because you have 0 gates, what does that mean? Does that mean we're always going to be closed or always going to be open? In this case, for pores, you're always going to be open. So you're going to have a model that looks somewhat similar to this. So there's always going to be an opening here in which molecules can flow through depending on their concentration gradient. Remember, we have to go down our concentration gradient from high to low. Awesome. Now we have our channels, and channels have 1 gate, like we said before. So it's going to look like this. And this gate can open, leading us to have molecules flowing down. And these gates are stimulated to be opened using electrical, chemical, or mechanical signals. Lastly, we have our carrier or our uniporter, and this one has 2 gates. So it's going to look a little bit as follows. You're going to have a bit of an opening like this, which can then be changed to something that looks a little bit along the lines of this. So you're going to have your input molecule flow through, and keep in mind, a big difference for this type of facilitated diffusion is that bigger molecules like glucose and other ions can flow through. So glucose, in this case, is a go. Great. So once we have this initial molecule flow through, this is what triggers the other side of the gate to open. So now, once this goes all the way through and reaches on this side of the gate will open and allows molecules to flow through the other way. So unlike simple diffusion, facilitated diffusion is not linear. Instead, it is logarithmic. The last type of transportation in our trifecta is going to be active transportation. So active transportation. Great. Unlike our previous methods of transportation, active transportation deals with molecules or ions going across their concentration gradient, but the other way around. So now we're going to be moving from low to high. In this case, we're going against the nature of the high to low reestablishing equilibrium, which means we're going to need our lovely friend ATP. We're going to need you. Awesome. So there's 2 types of active transportation. We have our primary and our secondary. So our primary consists of our pumps and ATPases. They're otherwise known as ATPases. And these use our ATP hydrolysis to move ions and molecules across their concentration gradient. A very common one we're going to be discussing later on in class is the sodium-potassium pump. And we also have secondary transportation, which utilizes transporting ions across the gradient by using another solute or molecule. So this is going to be co-transportation. Similar to our unitransporter, where we have two gates, this one is also going to be similar, but instead we're moving it against the concentration gradient. So remember, a big hallmark of active transportation itself is moving molecules against their concentration gradient. So for secondary, we have two gates, and we're going to need to use a co-transporter in order to see transportation against the gradient. Now, within this co-transportation, there's two specific types. We have our symporters, which are transporting two molecules in the same direction. And we also have our antiporters. And antiporters sound like the opposite of symporters, which means that we have one molecule going in one direction and another one going in another direction. So different directions. Now that we've gone through the main methods of transportation, let's talk about the way that these neurotransmitters are going through these methods of transportation, if that makes sense. And this is utilizing a vesicle. So our vesicles. I'm going to go through this pretty quickly because there isn't much I want to talk about that'll be too pertinent to this course. But there's two main portions of a vesicle that need to be discussed, and that is the dynamine and clathrin. I'm going to quickly draw a structure that represents this vesicle. I apologize for my lack of artistic ability, but bear with me for a little bit. So dynamine is a GTPase enzyme that mediates the molecules or ions going into a cell, so endocytosis. This is what creates this circular-like image that looks sort of like a soccer ball. So think soccer ball, GTPase, endocytosis. Clathrin. Clathrin is the protein that coats, clathrin coats the vesicle formed from endocytosis. So this grid-like pattern I just drew over here, this stuff, is our clathrin. So keywords to know is coats and endocytosis and pattern. That'll help you guys remember these two main parts of our neurotransmitter vesicle. So the last portion of this video that we're going to be discussing today is the snare complex. I'm going to be dividing this into three parts, and it's going to be as follows with the V snare, T snare, and synaptotagmin. I'm about to highlight or underline a letter in each of these so it helps everyone remember the function for each one. So the V snare is responsible for having our vesicle attached to our presynaptic membrane before it goes and fuses into our postsynaptic cell. So vesicle, this is where the V comes in. The vesicle binds to presynaptic membrane. T-snare. Our T-snare is when our vesicle fuses to the membrane. Vesicle fuses to membrane. Specifically, our target membrane. So not like our presynaptic membrane, this time it's going to be your target membrane. So the T here is important. Synaptotagmin. This is where our protein, which is attached to the vesicle, binds with calcium and makes it bind to the actual snare. So this is what pulls our vesicle into the membrane, into the active zone. So pulls vesicle to active zone. Like I said previously, I'm going to be attaching another set of figures to help describe the snare complex a little bit more in depth. But for the sake of this video, just remember these three main portions: the V snare, the T snare, and the synaptotagmin. There's a more in-depth name for each one of these, and I'm gonna write them down. But for the sake of this course, I do not think it's very necessary for everyone to memorize these. Synaptotaxin and synaptotagmin is already the scientific name. So one last portion to talk about in synaptotagmin, another way to remember this, another important fact to remember is that it requires a calcium, correct? And there's two T's in synaptotagmin, which can relate to the Ca2+, the 2+ charge that it has. So Ca2+, 2 T's. Awesome job, guys. Thanks so much for following along with this video. Please let me know if there's any way I can make anything more clear in the comments below, or if there's any figures or topics you guys would like me to go over in the future. And until then, my name is Aditya Prabhakar. Thank you so much.